Panmictic species. Pantala flavescens is considered as a global panmictic population. Panmictic population of Monostromal latissimum, a marine green algae, shows sympatric speciation in southwest Japanese islands. Although panmictic, the population is diversifying. Simpson's burrowing bee, a megalodocene, may be forced to aggregate in common mating areas due to uneven resource distribution in its harsh desert environment. 7. Pantala flavescens should be considered as a global panmictic population. 10. Related experiments and species. 1. Anguilla rostrata, or the American eel, exhibits panmixia throughout the entire species. This allows the eel to have phenotypic variation in their offspring and survive in a wide range of environmental conditions. 11.8. 2. In 2016, BMC Evolutionary Biology conducted a study on Pachygrapsis marmoratus, the marbled crab, marking them as panmictic species. This claimed that the crab's mating behavior is characterized by genetic differentiation due to geographic breaks across its distribution range and not panmixia. 12. 3. In a heterogeneous environment such as the forests of Oregon, United States, Douglas squirrels Tamiocyrus douglasii exhibit local patterns of adaptation. In a study conducted by Chaves 2014, a population along an entire transect was found to be panmictic. Traits observed in this study included skull shape, fur color, etc. 4. Swordfish based in the Indian Ocean, Zithias gladius, have been found to be a single panmictic population. A study carried out by Muths et al. 2013 found large spatial and temporal homogeneity in genetic structure satisfactory in order to consider the swordfish's singular panmictic population. See Gene flow. Population genetics. Gene flow, also known as migration and allele flow, is the transfer of genetic material from one population to another. If the rate of gene flow is high enough, then two populations will have equivalent allele frequencies and therefore can be considered a single effective population. It has been shown that it takes only one migrant per generation to prevent populations from diverging due to drift. Populations can diverge due to selection even when they are exchanging alleles, if the selection pressure is strong enough. 2. 3. Gene flow is an important mechanism for transferring genetic diversity among populations. Change the distribution of genetic diversity among populations by modifying allele frequencies, the proportion of members carrying a particular variant of a gene. High rates of gene flow can reduce the genetic differentiation between the two groups, increasing homogeneity. 4. For this reason, gene flow has been thought to constrain speciation and prevent range expansion by combining the gene pools of the groups, thus preventing the development of differences in genetic variation that would have led to differentiation and adaptation. 5. In some cases, dispersal resulting in gene flow may also result in the addition of novel genetic variants under positive selection to the gene pool of a species or population, adaptive introgression. 6. Gene flow is the transfer of alleles from one population to another population through immigration of individuals. There are a number of factors that affect the rate of gene flow between different populations. Genes are expected to be lower in species that have low dispersal or mobility, that occur in fragmented habitats, where there is long distances between populations, and when there are small population sizes. 7. 8. Mobility plays an important role in dispersal rate, as highly mobile individuals tend to have greater movement prospects. 9. Although animals are thought to be more mobile than plants, pollen and seeds may be carried great distances by animals, water or wind. When gene flow is impeded, there can be an increase in inbreeding, measured by the inbreeding coefficient F within a population. For example, many island populations have low rates of gene flow due to geographic isolation and small population sizes. The black-footed rock wallaby has several inbred populations that live on various islands off the coast of Australia. The population is so strongly isolated that lack of gene flow has led to high rates of inbreeding. 10. Measuring gene flow. The level of gene flow among populations can be estimated by observing the dispersal of individuals and recording their reproductive success. 4. 11. Direct method is only suitable for some types of organisms. More often indirect methods are used that infer gene flow by comparing allele frequencies among population samples. 4. The more genetically differentiated two populations are, the lower the estimate of gene flow, because gene flow has a homogenizing effect. Isolation of populations leads to divergence due to drift, while migration reduces divergence. 10. Measuring gene flow. The level of gene flow among populations can be estimated by observing the dispersal of individuals and recording their reproductive success. 4. 11. This direct method is only suitable for some types of organisms. More often indirect methods are used that infer gene flow by comparing allele frequencies among population samples. 1. 4. The more genetically differentiated two populations are, the lower the estimate of gene flow, because gene flow has a homogenizing effect. Isolation of populations leads to divergence due to drift, while migration reduces divergence. Gene flow can be measured by using the effective population size and the net migration rate per generation m. Using the approximation based on the island model, the effect of migration can be calculated for a population in terms of the degree of genetic differentiation. 12. This formula accounts for the proportion of total molecular marker variation among populations, averaged over loci. 13. When there is one migrant per generation, the inbreeding coefficient equals 0.2. However, when there is less than one migrant per generation, no migration, the inbreeding coefficient rises rapidly, resulting in fixation and complete divergence equals 1. The most common is less than 0.25. This means there is some migration happening. Measures of population structure range from 0 to 1. When gene flow occurs via migration, the deleterious effects of inbreeding can be ameliorated. 1. The formula can be modified to solve for the migration rate when is known. NM equals number of migrants. 1. Barriers to gene flow. Allopatric speciation. Examples of speciation affecting gene flow. Is blocked by physical barriers. This results in allopatric speciation, or a geographical isolation that does not allow populations of the same species to exchange genetic material. Physical barriers to gene flow are usually, but not always, natural. They may include impassable mountain ranges, oceans, or vast deserts. In some cases, they can be artificial, human-made barriers, such as the Great Wall of China, which has hindered the gene flow of native plant populations. These native plants, Ulmus pumila, demonstrated a lower prevalence of genetic differentiation than the plants Videx negundo, Ziziphus jujuba, Heteropappus hispidus, and Prunus armeniaca, whose habitat is located on the opposite side of the Great Wall of China where Ulmus pumila grows. 14. This is because Ulmus pumila has wind pollination as its primary means of propagation, and the latter plants carry out pollination through insects. 4. Examples of the same species which grow on either side have been shown to have developed genetic differences, because there is little to no gene flow to provide recombination of the gene pools. Sympatric speciation. Barriers to gene flow need not always be physical. Sympatric speciation happens when new species from the same ancestral species arise along the same range. This is often a result of a reproductive barrier. For example, palm species of Howea found on Lord Howe Island were found to have substantially different flowering times correlated with soil preference, resulting in a reproductive barrier inhibiting gene flow. 50 species can live in the same environment, yet show very limited gene flow due to reproductive barriers, fragmentation, specialist pollinators, or limited hybridization or hybridization yielding unfit hybrids. A cryptic species is a species that humans cannot tell is different without the use of genetics. Moreover, gene flow between hybrid and wild populations can result in loss of genetic diversity via genetic pollution, assortative mating and outbreeding. In human populations, genetic differentiation can also result from endogamy, due to differences in caste, ethnicity, customs and religion. Human-assisted gene flow. Genetic rescue. Gene flow can also be used to assist species which are threatened with extinction. When a species exists in small populations, there is an increased risk of inbreeding and greater susceptibility to loss of diversity due to drift. These populations can benefit greatly from the introduction of unrelated individuals 11 who can increase diversity 16 and reduce the amount of inbreeding and potentially increase population size. 17 is demonstrated in the lab with two bottleneck strains of Drosophila melanogaster, in which crosses between the two populations reversed the effects of inbreeding and led to greater chances of survival. Gene flow can also be used to assist species which are threatened with extinction. When a species exists in small populations, there is an increased risk of inbreeding and greater susceptibility to loss of diversity due to drift. These populations can benefit greatly from the introduction of unrelated individuals 11 who can increase diversity 16 and reduce the amount of inbreeding and potentially increase population size. 17. This was demonstrated in the lab with two bottleneck strains of Drosophila melanogaster, in which crosses between the two populations reversed the effects of inbreeding and led to greater chances of survival in not only one generation but two. 18. Genetic pollution. Activities such as movement of species and modification of landscape can result in genetic pollution, hybridization, introgression and genetic swamping. These processes can lead to homogenization or replacement of local genotypes as a result of either a numerical and/or fitness advantage of introduced plant or animal. Non-native species can threaten native plants and animals with extinction by hybridization and introgression, either through purposeful introduction by humans or through habitat modification, bringing previously isolated species into contact. These phenomena can be especially detrimental for rare species coming into contact with more abundant ones, which can occur between island and mainland species. Interbreeding between the species can cause a swamping of the rare species gene pool, creating hybrids that supplant the native stock. A direct result of evolutionary forces such as natural selection, as well as genetic drift, which lead to the increasing prevalence of advantageous traits and homogenization. The extent of this phenomenon is not always apparent from outward appearance alone. While a degree of gene flow occurs in the course of normal evolution, hybridization with or without introgression may threaten a rare species' existence. 21. For example, the mallard is an abundant species of duck that interbreeds readily with a wide range of other ducks and poses a threat to the integrity of some species. 22. Urbanization. There are two main models for how urbanization affects gene flow of urban populations. The first, habitat fragmentation, also called urban fragmentation, in which alterations to the landscape that disrupt or fragment the habitat decrease genetic diversity. The second is called the urban facilitation model, and suggests that in some populations, gene flow is enabled by anthropogenic changes to the landscape. Urban facilitation of gene flow connects populations, reduces isolation, and increases gene flow into an area which would otherwise not have this specific genome composition. 23. Urban facilitation can occur in many different ways, but most of the mechanisms include bringing previously separated species into contact, either directly or indirectly. Altering a habitat through urbanization. The first is through habitat fragmentation, also called urban. There are two main models for how urbanization affects gene flow of urban populations. The first is through habitat fragmentation, also called urban fragmentation. A cryptic species is a species that humans cannot tell is different without the use of genetics. Moreover, gene flow between hybrid and wild populations can result in loss of genetic diversity via genetic pollution, assortative mating and outbreeding. In human populations, genetic differentiation can also result from endogamy, due to differences in caste, ethnicity, customs and religion. Human-assisted gene flow. Genetic rescue. Gene flow can also be used to assist species which are threatened with extinction. When species exist in small populations, there is an increased risk of inbreeding and greater susceptibility to loss of diversity due to drift. These populations can benefit greatly from the introduction of unrelated individuals 11 who can increase diversity 16 and reduce the amount of inbreeding and potentially increase population size. 7 is demonstrated in the lab with two bottleneck strains of Drosophila melanogaster, in which crosses between the two populations reverse the effects of inbreeding and led to greater chances of survival in not only one generation but two. 18. Genetic pollution. Human activities such as movement of species and modification of landscape can result in genetic pollution, hybridization, introgression and genetic swamping. Processes can lead to homogenization or replacement of local genotypes as a result of either a numerical and/or fitness advantage of introduced plant or animal. 19. Non-native species can threaten native plants and animals with extinction by hybridization and introgression either through purposeful introduction by humans or through habitat modification, bringing previously isolated species into contact. These phenomena can be especially detrimental for rare species coming into contact with more abundant ones, which can occur between island and mainland species. Interbreeding between the species can cause a swamping of the rare species gene pool, creating hybrids that supplant the native stock. This is a direct result of evolutionary forces such as natural selection, as well as genetic drift, which lead to the increasing prevalence of advantageous traits and homogenization. The extent of this phenomenon is not always apparent from outward appearance alone. While some degree of gene flow occurs in the course of normal evolution, hybridization with or without introgression may threaten a rare species' existence. 21. For example, the mallard is an abundant species of duck that interbreeds readily with a wide range of other ducks and poses a threat to the integrity of some species. 22. Urbanization. There are two main models for how urbanization affects gene flow of urban populations. The first is through habitat fragmentation, also called urban fragmentation, in which alterations to the landscape that disrupt or fragment the habitat decrease genetic diversity. This is called the urban facilitation model, and suggests that in some populations, gene flow is enabled by anthropogenic changes to the landscape. Urban facilitation of gene flow connects populations, reduces isolation, and increases gene flow into an area which would otherwise not have this specific genome composition. 23. Urban facilitation can occur in many different ways, but most of the mechanisms include bringing previously separated species into contact, either directly or indirectly. Altered urbanization will cause habitat fragmentation, but could also potentially disrupt barriers and create a pathway, or corridor, that can connect two formerly separated species. The effectiveness of this depends on individual species' dispersal abilities and adaptiveness to different environments to use anthropogenic structures to travel. Even climate change is another mechanism by which southern-dwelling animals might be forced northward towards cooler temperatures, where they could come into contact with other populations not previously in their range. More directly, humans are known to introduce non-native species into new environments, which could lead to hybridization of similar species. 24. This urban facilitation model was tested on a human health pest, the western black widow spider, Latrodectus hesperus. A study by Michael collected genome-wide single nucleotide polymorphism variation data in urban and rural spider populations and found evidence for increased gene flow in urban western black widow spiders compared to rural populations. In addition, the genome of these spiders was more similar across rural populations than it was for urban populations, suggesting increased diversity, and therefore adaptation, in the urban populations of the western black widow spider. Typically, urban spiders are larger, darker, and more aggressive, which could lead to increased survival in urban environments. These demonstrate support for urban facilitation, as these spiders are actually able to spread and diversify faster across urban environments than they would in a rural one. However, it is also an example of how urban facilitation, despite increasing gene flow, is not necessarily beneficial to an environment, as western black widow spiders have highly toxic venom and therefore pose risks for human health. 25. Another example of urban facilitation is that of migrating bobcats, Lynx rufus, in the northern U.S. and southern Canada. A study by et al. Sequenced 14 different microsatellite loci in bobcats across the Great Lakes region, and found that longitude affected the interaction between anthropogenic landscape alterations and bobcat population gene flow. While rising global temperatures push bobcat populations into northern territory, increased human activity also enables bobcat migration northward. The increased human activity brings increased roads and traffic, but also increases road maintenance, plowing, and snow compaction, inadvertently clearing a path for bobcats to travel by. The anthropogenic influence on bobcat migration pathways is an example of urban facilitation via opening up a corridor for gene flow. How, in the bobcat's southern range, an increase in roads and traffic is correlated with a decrease in forest cover, which hinders bobcat population gene flow through these areas. Subdirectly, the movement of bobcats northward is caused by human-driven global warming, but is also enabled by increased anthropogenic activity in northern ranges that make these habitats more suitable to bobcats. 26. Consequences of urban facilitation vary from species to species. Effects of urban facilitation can occur when increased gene flow enables better adaptation and introduces beneficial alleles, and would ideally increase biodiversity. Implications for conservation. For example, urban facilitation benefits an endangered species of tarantula and could help increase the population size. Negative effects would occur when increased gene flow is maladaptive and causes a loss of beneficial alleles. In the worst-case scenario, this would lead to genomic extinction through a hybrid swarm. It is important to note that in the scheme of overall ecosystem health and biodiversity, urban facilitation is not necessarily beneficial, and generally applies to urban adapter pests. Examples of this include the previously mentioned western black widow spider, and also the cane toad, which was able to use roads by which to travel and overpopulate Australia. 23. Gene flow between species. Horizontal gene transfer. Horizontal gene transfer, HGT, transfer of genes between organisms in a manner other than traditional reproduction, either through transformation, direct uptake of genetic material by a cell from its surroundings, conjugation, transfer of genetic material between two bacterial cells in direct contact, transduction, injection of foreign DNA by a bacteriophage virus into the host cell, or GTA-mediated transduction, transfer by a virus-like element produced by a bacterium. 27-28. Virus. Horizontal gene transfer. Horizontal gene transfer, HG, transfer of genes between organisms in a manner other than traditional reproduction, either through transformation, direct uptake of genetic material by a cell from its surroundings, conjugation, transfer of genetic material between two bacterial cells in direct contact, transduction, injection of foreign DNA by a bacteriophage virus into the host cell, or GTA-mediated transduction, transfer by a virus-like element produced by a bacterium. 27-28. Viruses can transfer genes between species. 29. Bacteria can incorporate genes from dead bacteria, exchange genes with living bacteria, and can exchange plasmids across species boundaries. Sequence comparisons suggest recent horizontal transfer of many genes among diverse species, including across the boundaries of phylogenetic domains. Determining the phylogenetic history of a species cannot be done conclusively by determining evolutionary trees for single genes. 31. Biologist Gogarden suggests, the original metaphor of a tree no longer fits the data from recent genome research. Biologists should use the metaphor of a mosaic to describe the different histories combined in individual genomes and use the metaphor of an intertwined net to visualize the rich exchange and cooperative effects of horizontal gene transfer. 32. Using single genes as phylogenetic markers, it is difficult to trace organismal phylogeny in the presence of HGT. Combined coalescence model of cladogenesis with rare HGT events suggests there was no single last common ancestor that contained all of the genes ancestral to those shared among the three domains of life. Each contemporary molecule has its own history and traces back to an individual molecule's ancestor. However, these molecular ancestors were likely to be present in different organisms at different times. 33. Hybridization. When a species has a sister species and breeding capabilities are possible due to the removal of previous barriers or through introduction due to human intervention, species can hybridize and exchange genes and corresponding traits. 34. This exchange is not always clear-cut, for sometimes the hybrids may look identical to the original species phenotypically, but upon testing the mtDNA it is apparent that hybridization has occurred. Hybridization also occurs because some traits in DNA are more readily exchanged than others, and this is a result of selective pressure or the absence thereof that allows for easier transaction. In instances the introduced species begins to replace the native species becomes threatened and the biodiversity is reduced, thus making this phenomenon negative rather than a positive case of gene flow that augments genetic diversity. 35. Integration is the replacement of one species' alleles with that of the invader species. It is important that hybrids are sometimes less fit than their parental generation. 36. And as a result is a closely monitored genetic issue, as the ultimate goal in conservation genetics is to maintain the genetic integrity of a species and preserve biodiversity. Examples. Marine iguana of the Galapagos Islands evolved via allopatric speciation, through limited gene flow and geographic isolation. Gene flow can greatly enhance the fitness of a population. It can also have negative consequences depending on the population and the environment in which they reside. The effects of gene flow are context-dependent. Fragmented population. Fragmented landscapes such as the Galapagos Islands are an ideal place for adaptive radiation to occur as a result of differing geography. Darwin's finches likely experienced allopatric speciation in some part due to differing geography, but that does not explain why we see so many different kinds of finches on the same island. This is due to adaptive radiation, or the evolution of varying traits in light of competition for resources. Gene flow moves in the direction of what resources are abundant at a given time. 37. Population. The marine iguana is an endemic species of the Galapagos Islands, but it evolved from a mainland ancestor of land iguana. Due geographic isolation, gene flow between the two species was limited and differing environments caused the marine iguana to evolve in order to adapt to the island environment. For instance, they are the only iguana that has evolved the ability to swim. Human populations. In Europe, Homo sapiens interbred with Neanderthals, resulting in gene flow between these populations. 38. This gene flow has resulted in Neanderthal alleles in modern European population. 39. Two theories exist for the human evolution throughout the world. This is a multi-regional model in which modern human variation is seen as a product of radiation of Homo erectus out of Africa, after which local differentiation led to the establishment of regional population as we see them now. 40-41. Gene flow plays an important role in maintaining a grade of similarities and preventing speciation. The single origin theory assumes that there was a common ancestral population originating in Africa of Homo sapiens which already displayed the anatomical characteristics we see today. This theory minimizes the amount of parallel evolution that is needed. 41. Butterflies. Comparisons between sympatric and allopatric populations of Heliconius melpomene, H. sydno, and H. timeretta revealed a genome-wide trend of increased shared variation in sympatry, indicative of pervasive interspecific gene flow. 42. Mediated gene flow. The captive genetic management of threatened species is the only way in which humans attempt to induce gene flow in ex situ situation. The giant panda, which is part of an international breeding program in which genetic materials are shared between zoological organizations in order to increase genetic diversity in the small populations. As a result of low reproductive success, artificial insemination with fresh, frozen thawed sperm was developed, which increased cub survival rate. A 2014 study found that high levels of genetic diversity and low levels of inbreeding were estimated in the breeding centers. 43. Two populations of monkey flowers were found to use different pollinators. One example is the giant panda. Human-mediated gene flow. The captive genetic management of threatened species is the only way in which humans attempt to induce gene flow in ex situ situation. One example is the giant panda, which is part of an international breeding program in which genetic materials are shared between zoological organizations in order to increase genetic diversity in the small populations. As a result of low reproductive success, artificial insemination with fresh, frozen thawed sperm was developed, which increased cub survival rate. A 2014 study found that high levels of genetic diversity and low levels of inbreeding were estimated in the breeding centers. 43. Plants. Populations of monkey flowers were found to use different pollinators, bees and hummingbirds, that limited gene flow, resulting in genetic isolation, eventually producing two different species, Mamulus lewisi and Mamulus cardinalis. 44. Sika deer. Sika deer were introduced into Western Europe, and they reproduce easily with the native red deer. This location of Sika deer has led to introgression, and there are no longer pure red deer in the region, and all can be classified as hybrids. 45. Quail. Bobwhite quail were translocated from the southern part of the United States to Ontario in order to increase population numbers and game for hunting. The hybrids that resulted from this translocation was less fit than the native population and were not adapted to survive the northern winters. Fixation index. The fixation index, FST, is a measure of population differentiation due to genetic structure. It is frequently estimated from genetic polymorphism data, such as single nucleotide polymorphisms, SNP, or microsatellites. Developed as a special case of Wright's F statistics, it is one of the most commonly used statistics in population genetics. Its values range from 0 to 1, with 0 being no differentiation and 1 being complete differentiation. FST values between European populations. Interpretation. This comparison of genetic variability within and between populations is frequently used in applied population genetics. The values range from 0 to 1. A 0 value implies complete panmixia, that is, that the two populations are interbreeding freely. A 1 implies that all genetic variation is explained by the population structure, and that the two populations do not share any genetic diversity. For idealized models such as Wright's finite island model, FST can be used to estimate migration rates. Under that model, the migration rate is comma, where m is the migration rate per generation, and is the mutation rate per generation. 1. The interpretation of FST can be difficult when the data analyzed are highly polymorphic. In this case, the probability of identity by descent is very low and FST can have an arbitrarily low upper bound, which might lead to misinterpretation of the data. Also, strictly speaking, FST is not a distance in the mathematical sense, as it does not satisfy the triangle inequality. Definition. Two of most commonly used definitions for FST at a given locus are based on 1, the variance of allele frequencies among populations, and on 2, the probability of identity by descent. If is the average of an allele in the total population, is the variance in the frequency of the allele among different subpopulations, weighted by the sizes of the subpopulations, and is the variance of the allelic state in the total population, FST is defined as 2. This definition illustrates that FST measures the amount of genetic variance that can be explained by population structure. This can be the fraction of total diversity that is not a consequence of the average diversity within subpopulations, where diversity is measured by the probability that two randomly selected alleles are different, namely, if allele frequency in the TH population is in the relative size of the TH population is, then alternatively, 3. Where is the probability of identity by descent of two individuals given that the two individuals are in the same subpopulation, and is the probability that two individuals from the total population are identical by descent? Using this definition, FST can be interpreted as measuring how much closer two individuals from the same subpopulation are, compared to the total population. If the mutation rate, this interpretation can be made more explicit by linking the probability of identity by descent to coalescent times. Let T0 and T denote the average time to coalescence for individuals from the same subpopulation and the total population, respectively. Then, this has the advantage that the expected time to coalescence can easily be estimated from genetic data, which led to the development of various estimators for FST. Estimation. In practice, none of the quantities used for the definitions can be easily measured. As a consequence, various estimators have been proposed. A particularly simple estimator applicable to DNA sequence data is 4. Where present the average number of pairwise differences between two individuals sampled from different subpopulations or from the same subpopulation. Closing parenthesis dot. The average pairwise difference within a population can be calculated as the sum of the pairwise differences divided by the number of pairs. However, this estimator is biased when sample sizes are small or if they vary between populations. Therefore, more elaborate methods are used to compute FST in practice. Two of the most widely used procedures are the estimator by Weir and Cockerham, 1984-5, or performing an analysis of molecular variance. A list of implementations is available at the end of this article. FST in humans. FST values in selected populations. FST values depend strongly on the choice of populations. Closely related ethnic groups, such as the Danes versus the Dutch, or the Portuguese versus the Spaniards, show values significantly below 1%, indistinguishable from panmixia. Within Europe, the most divergent ethnic groups have been found to have values of the order of 7%, Sami versus Sardinians. Values are found if highly divergent homogeneous groups are compared. The highest such value found was at close to 46%, between Budi and Papuans. 6. A genetic distance of 0.125 implies that kinship between unrelated individuals of the same ancestry relative to the world population is equivalent to kinship between half-siblings in a randomly mating population. Implies that if a human from a given ancestral population has a mixed half-sibling, that human is closer genetically to an unrelated individual of their ancestral population than to their mixed half-sibling. 7. Genetic distances in human populations. Autosomal genetic distances based on classical markers. In the Theory and Geography of Human Genes, 1994, Cavalli-Sforza, Manozzi and Piazza provide some of the most detailed and comprehensive estimates of genetic distances between human populations, within and across continents. Their initial database contains 76,676 gene frequencies, using 120 blood polymorphisms, corresponding to 6,633 samples in different locations. By culling and pooling such samples, they restrict their analysis to 491 populations. The 42 world populations used by the authors and their reported FSTs. They focus on Aboriginal populations that were at their present location at the end of the 15th century when the Great European Migrations began. Studying genetic difference at the world level, the number is reduced to 42 representative populations, aggregating subpopulations characterized by a high level of genetic similarity. For these 42 populations, Cavalli-Sforza and co-authors report bilateral distances computed from 120 alleles. Among this set of 42 world populations, the greatest genetic distance observed is between Budi Pygmies and Papua New Guineans, where the FST distance is 0.4573, while the smallest genetic distance, 0.0021, is between the Danish and the English. When considering more disaggregated data for 26 European populations, the smallest genetic distance, 0.0009, is between the Dutch and the Danes, and the largest, 0.0667, is between the Lapps and the Sardinians. Mean genetic distance among the 861 available pairings of the 42 selected populations was found to be 0.1338. Polygen. A polygen is a member of a group of non-epistatic genes that interact additively to influence a phenotypic trait, thus contributing to multiple gene inheritance, polygenic inheritance, multigenic inheritance, quantitative inheritance, 1, a type of non-Mendelian inheritance, as opposed to single gene inheritance, which is the core notion of Mendelian inheritance. The term homologous is usually used to refer to a hypothetical gene, as it is often difficult to distinguish the effect of an individual gene from the effects of other genes in the environment on a particular phenotype. Advances in statistical methodology and high-throughput sequencing are, however, allowing researchers to locate candidate genes for the trait. In the case that such a gene is identified, it is referred to as a quantitative trait locus, QTL. These genes are generally pleiotropic as well. The genes that contribute to type 2 diabetes are thought to be mostly polygens. In July 2016, scientists reported identifying a set of 355 genes from the last universal common ancestor, LUCA, of all organisms living on Earth. 3. Traits with polygenic determinism correspond to the classical quantitative characters, as opposed to the qualitative characters with monogenic or oligogenic determinism. Essence instead of two options, such as freckles or no freckles, there are many variations, like the color of skin, hair, or even eyes. Overview. Polygenic locus is any individual locus which is included in the system of genes responsible for the genetic component of variation in a quantitative polygenic character. Allelic substitutions contribute to the variance in a specified quantitative character. Polygenic locus may be either a single or complex genetic locus in the conventional sense, i.e., either a single gene or closely linked block of functionally related genes. 4. In modern sense, the inheritance mode of polygenic patterns is called polygenic inheritance, whose main properties may be summarized as follows. 1. Most metric and meristic traits are controlled by a number of genetic loci. 2. Main mode of non-allelic genes interaction in corresponding gene series is addition of mainly small particular allele contributions. In modern sense, the inheritance polygenic locus is any individual locus which is included in the system of genes responsible for the genetic component of variation in a quantitative polygenic character. Allelic substitutions contribute to the variance in a specified quantitative character. Polygenic locus may be either a single or complex genetic locus in the conventional sense, i.e., either a single gene or closely linked block of functionally related genes. 4. In modern sense, the inheritance mode of polygenic patterns is called polygenic inheritance, whose main properties may be summarized as follows. 1. Most metric and meristic traits are controlled by a number of genetic loci. 2. Main mode of non-allelic genes interaction in corresponding gene series is addition of mainly small particular allele contributions. 3. The allelic substitution at each of the segregating genes are usually relatively small and interchangeable, which results that identical phenotype may be displayed by a great variety of genotypes. 4. The phenotypic expression of the polygenic characters is undergoing considerable modification by environmental influence. 5. Polygenic characters show a continuous rather than discontinuous distribution. 6. Balanced and polygenic inheritance in a population contain a great deal of potential genetic variability in the heterozygous condition and released by small increments through genetic recombination between linked polygens. 5, 6, 7, 8. Inheritance. Polygenic inheritance occurs when one characteristic is controlled by two or more genes. Often the genes are large in quantity but small in effect. Examples of human polygenic inheritance are height, skin color, eye color and weight. Polygens exist in other organisms, as well. Drosophila, for instance, display polygyny with traits such as wing morphology, 10 bristle count 11, and many others. Trait distribution. The frequency of the phenotypes of these traits generally follows a normal continuous variation distribution pattern. This results from the many possible allelic combinations. When the values are plotted, a bell-shaped, normal, curve is obtained. The mode of the distribution represents the optimal, or fittest, phenotype. The more genes are involved, the smoother the estimated curve, which follows from the central limit theorem. It implies that traits such as height that are both highly heritable and normally distributed are necessarily polygenic. In other words, the fact that human height follows a smooth bell curve implies that there can be no single gene, or even small cluster of genes, that control height under ordinary circumstances. However, in this model all genes must code for alleles with additive effects. This assumption is unrealistic as many genes display epistasis effects which can have unpredictable effects on the distribution of outcomes, especially when looking at the distribution on a fine scale. 12. Mapping polygens. Example of a genome-wide scan for QTL of osteoporosis. Traditionally, mapping polygens requires statistical tools available to help measure the effects of polygens as well as narrow in on single genes. One of these tools is QTL mapping. QTL mapping utilizes a phenomenon known as linkage disequilibrium by comparing known marker genes with correlated phenotypes. Often, researchers will find a large region of DNA, called a locus, that accounts for a significant amount of the variation observed in the measured trait. This locus will usually contain a large number of genes that are responsible. A new form of QTL has been described as expression QTL, eQTL. eQTLs regulate the amount of expressed mRNA, which in turn regulates the amount of protein within the organism. 13. An interest of statistical geneticists using QTL mapping is to determine the complexity of the genetic architecture underlying a phenotypic trait. For example, they may be interested in knowing whether a phenotype is shaped by many independent loci, or by a few loci, and do those loci interact. This can provide information on how the phenotype may be evolving. Complex traits. Complex traits are phenotypes that are controlled by two or more genes and do not follow Mendel's law of dominance. They may have a range of expression which is typically continuous. Both environmental and genetic factors often impact the variation in expression. Human height is a continuous trait, meaning that there is a wide range of heights. There are an estimated 50 genes that affect the height of a human. Environmental factors, like nutrition, also play a role in a human's height. Other examples of complex traits include crop yield, plant color, and many diseases including diabetes and Parkinson's disease. One major goal of genetic research today is to better understand the molecular mechanisms through which genetic variants act to influence complex traits. Complex traits are also known as polygenic traits and multigenic traits. The size of a tomato is one example of a complex trait. The X traits, which are far more common than Mendelian traits, represented a significant challenge to the acceptance of Mendel's work. Modern understanding has three categories of complex traits: quantitative, meristic, and threshold. These traits have been studied on a small scale with observational techniques like twin studies. They are also studied with statistical techniques like quantitative trait loci (QTL) mapping, and genome-wide association studies (GWAS) on a large scale. The overall goal of figuring out how genes interact with each other in the environment and how those interactions can lead to variation in a trait is called genetic architecture. History. Mendel's work on inheritance was rediscovered in 1900. Scientists debated whether Mendel's laws could account for the continuous variation observed for many traits. One group of biometricians argued that continuous traits such as height were largely heritable, but could not be explained by the inheritance of single Mendelian genetic factors. Work published by Ronald Fisher in 1919 mostly resolved debate by demonstrating that the variation in continuous traits could be accounted for if multiple such factors contributed additively to each trait. However, the number of genes involved in such traits remained undetermined. Until recently, genetic loci were expected to have moderate effect sizes and each explain several percent of heritability. 3. After the conclusion of the Human Genome Project in 2001, it seemed that the sequencing and mapping of many individuals would soon allow for a complete understanding of traits' genetic architectures. However, variants through genome-wide association studies (GWASs) accounted for only a small percentage of predicted heritability. For example, while height is estimated to be 80–90% heritable, early studies only identified variants accounting for 5% of this heritability. 4. Later research showed that most mean heritability could be accounted for by common variants missed by GWASs because their effect sizes fell below significance thresholds. A smaller percentage is accounted for by rare variants with larger effect sizes, although in certain traits such as autism, rare variants play a more dominant role. While many genetic factors involved in complex traits have been identified, determining their specific contributions to phenotypes, specifically, the molecular mechanisms through which they act, remains a major challenge. 8. Types of complex traits. Quantitative traits. Quantitative traits have phenotypes that are expressed on continuous ranges. 9. They have many different genes that impact the phenotype, with differing effect sizes. 10. Many of these traits are somewhat heritable. For example, height is estimated to be 60–80% heritable, however, other quantitative traits have varying heritability. 11. Meristic traits. Meristic traits have phenotypes that are described by whole numbers. An example is the rate chickens lay eggs. A chicken can lay 1, 2, or 5 eggs a week, but never half an egg. Environment can also impact expression, as chickens will not lay as many eggs depending on the time of year. 12. Threshold traits. Threshold traits have phenotypes that have limited expressions, usually 2. It is a complex trait because multiple genetic and environmental factors impact the phenotype. 14. The phenotype before the threshold is referred to as normal or absent, and after the threshold is lethal or present. These traits are often examined in a medical context, because many diseases exhibit this pattern or similar. 9. An example of this is type 2 diabetes. The phenotype is either normal, healthy or lethal, diseased. 15. Methods for finding complex traits. Twin studies. Studies is an observational test using monozygotic twins and dizygotic twins, preferably same-sex. They are used to figure out the environmental influence on complex traits. Monozygotic twins in particular are estimated to share 100% of their DNA with each other, so any phenotypic differences should be caused by environmental influences. 2. QTL mapping. Many complex traits are genetically determined by quantitative trait loci (QTL). Quantitative trait loci analysis can be used to find regions on the genome sequence that are associated with a complex trait. To find these regions, researchers will select a trait of interest and take a group of individuals of a species with varying expressions of this trait. They will label the individuals as founding parents and attempt to measure the trait. This can be difficult as most traits do not have a direct cutoff point. Researchers will then genotype the parents using molecular markers such as SNPs or RFLPs. These act as signposts pointing to an area of where the genes associated with a trait are. From there, the parents are crossed to produce offspring. These offspring are then made to produce new offspring, but who they breed with can vary. 17. They can either reproduce with their siblings, with themselves different from asexual reproduction, or backcross. 17. They can either reproduce with their siblings, with themselves different from asexual reproduction, or backcross. 18. After this, a new generation is produced that are more genetically diverse. This is due to recombination. The phenotype of this new generation are measured and compared with the molecular markers to identify which alleles are associated with the trait. 19. This does not mean there is a direct causal relationship between these regions in the trait, but it does give insight that there are genes that do have some relationship with the trait and reveals where to look in future research. GWAS. A genome-wide association study (GWAS) is a technique used to find gene variants linked to complex traits. A GWAS is done with populations that mate randomly because all the genetic variants are tested at once. Then researchers can compare the different alleles at a locus. It is similar to QTL mapping. 20. The most common setup for a GWAS is a case study which creates two populations, one with the trait we are looking at and one without the trait. In populations, researchers will map every subject's genome and compare them to find different variants in the SNPs between the two populations. Both populations should have similar environmental backgrounds. GWAS is only looking at the DNA and does not include differences that would be caused by environmental factors. 2. A Manhattan plot showing genome association with microcirculation. Statistical test, such as a chi-squared, is used to find if there is association with the trait in each of the SNPs tested. Statistical test produces a p-value which the researcher will use to conclude if the SNP is significant. This p-value cutoff can range from being a higher number or a lower number at the researcher's discretion. The data can then be visualized in a Manhattan plot which takes the log p-value, so all the significant SNPs are at the top of the graph. 21-22. Genetic architecture. Genetic architecture is an overall explanation of all the genetic factors that play a role in a complex trait and exists as the core foundation of quantitative genetics. With the use of mathematical models and statistical analysis, like GWAS, researchers can determine the number of genes affecting a trait as well as the level of influence each gene has on the trait. This isn't always easy as the architecture of one trait can be different between two separate populations of the same species. 16. This can be due to the fact that both populations live in different environments. Differing environments can lead to different interactions between genes and the environment, changing the architecture of both populations. 23. With rapid increases in available genetic data, researchers have begun to characterize the genetic architecture of complex traits better. One has been the observation that most loci identified in GWASs are found in non-coding regions of the genome. Therefore, instead of directly altering protein sequences, such variants likely affect gene regulation. 24. To understand the effects of these variants, QTL mapping has been employed to examine data from each step of gene regulation. For example, mapping RNA sequencing data can help determine the effects of variants on mRNA expression levels, which then presumably affect the numbers of proteins translated. A comprehensive analysis of keys involved in various regulatory steps, promoter activity, transcription rates, mRNA expression levels, translation levels, and protein expression levels, showed that high proportions of QTLs are shared, indicating that regulation behaves as a sequential ordered cascade, with variants affecting all levels of regulation. Many of these variants act by affecting transcription factor binding and other processes that alter chromatin function, steps which occur before and during RNA transcription. 25. To determine the functions of these variants, researchers have largely focused on identifying key genes, pathways, and processes that drive complex trait behavior. An inherent assumption has been that the most statistically significant variants have the greatest impact on traits because they act by affecting these key drivers. 26. For example, one study hypothesizes that there exist rate-limiting genes pivotal to the function of gene regulatory networks. 27. Other studies have identified the functional impacts of key genes and mutations on disorders, including autism and schizophrenia. 7. 20. A 2017 analysis by Boyle et al. Argues that while genes which directly impact complex traits do exist, regulatory networks are so interconnected that any expressed gene affects the functions of these core genes. This idea is called the omnigenic hypothesis. These peripheral genes each have small effects. Their combined impact far exceeds the contributions of core genes themselves. To support the hypothesis play a smaller than expected role, the authors describe three main observations. The heritability for complex traits is spread broadly, often uniformly, across the genome. Genetic effects do not appear to be mediated by cell type specific function, and genes in the relevant functional categories only modestly contribute more to heritability than other genes. One alternative to the omnigenic hypothesis is the idea that peripheral genes act not by altering core genes but by altering cellular states, such as the speed of cell division or hormone response. Phenotypic plasticity. Plasticity refers to some of the changes in an organism's behavior, morphology and physiology in response to a unique environment. 1. The way in which organisms cope with environmental variation. Phenotypic plasticity encompasses all types of environmentally induced changes, e.g. morphological, physiological, behavioral, phenological, that may or may not be permanent throughout an individual's lifespan. 3. The term was originally used to describe developmental effects on morphological characters, but is now more broadly used to describe all phenotypic responses to environmental change, such as acclimatization, as well as learning. 3. The special case when differences in environment induce discrete phenotypes is termed polyphenism. Phenotypic plasticity is the ability of one genotype to produce more than one phenotype when exposed to different environments. Each line here represents a genotype. Horizontal lines show that the phenotype is the same in different environments. Slanted lines show that there are different phenotypes in different environments, and thus indicate plasticity. Generally, phenotypic plasticity is more important for immobile organisms, e.g. plants, than mobile organisms, e.g. most animals, as mobile organisms can often move away from unfavorable environments. Nevertheless, mobile organisms also have at least some degree of plasticity in at least some aspects of the phenotype. 2. Mobile organism with substantial phenotypic plasticity is a Cyrthosyphon of the aphid family, which exhibits the ability to interchange between asexual and sexual reproduction, as well as growing wings between generations when plants become too populated. 5 water fleas Daphnia magna have shown both phenotypic plasticity and the ability to genetically evolve to deal with the heat stress of warmer, urban pond waters. 2. Examples. Phenotypic plasticity. Phenotypic plasticity refers to some of the changes in an organism's behavior, morphology and physiology in response to a unique environment. 1. 2. Fundamental way in which organisms cope with environmental variation. Phenotypic plasticity encompasses all types of environmentally induced changes, e.g. morphological, physiological, behavioral, phenological, that may or may not be permanent throughout an individual's lifespan. 3. The term was originally used to describe developmental effects on morphological characters, but is now more broadly used to describe all phenotypic responses to environmental change, such as acclimatization, as well as learning. 3. The special case when differences in environment induce discrete phenotypes is termed polyphenism. There is no previous article to read. Phenotypic plasticity. Phenotypic plasticity refers to some of the changes in an organism's behavior, morphology and physiology in response to a unique environment. 1. 2. Way in which organisms cope with environmental variation. Phenotypic plasticity encompasses all types of environmentally induced changes, e.g. morphological, physiological, behavioral, phenological, that may or may not be permanent throughout an individual's lifespan. 3. The term was originally used to describe developmental effects on morphological characters, but is now more broadly used to describe all phenotypic responses to environmental change, such as acclimatization, as well as learning. 3. The special case when differences in environment induce discrete phenotypes is termed polyphenism. Phenotypic plasticity is the ability of one genotype to produce more than one phenotype when exposed to different environments. Each line here represents a genotype. Horizontal lines show that the phenotype is the same in different environments. Slanted lines show that there are different phenotypes in different environments, and thus indicate plasticity. Generally, phenotypic plasticity is more important for immobile organisms, e.g. plants, than mobile organisms, e.g. most animals, as mobile organisms can often move away from unfavorable environments. 4. Nevertheless, mobile organisms also have at least some degree of plasticity in at least some aspects of the phenotype. 2. One mobile organism with substantial phenotypic plasticity is a Cyrthosyphon pisum of the aphid family, which exhibits the ability to interchange between asexual and sexual reproduction, as well as growing wings between generations when plants become too populated. 5. Water fleas Daphnia magna have shown both phenotypic plasticity and the ability to genetically evolve to deal with the heat stress of warmer, urban pond waters. 2. Examples. Plants. Phenotypic plasticity of sorghum flowering time evaluated from 7 environments. The hydrothermal time, a performance-independent index, quantifies the relevant environmental input and enables a systematic framework for modeling, explaining, and predicting phenotypic values under natural conditions. 6. Phenotypic plasticity includes the timing of transition from vegetative to reproductive growth stage, the allocation of more resources to the roots in soils that contain low concentrations of nutrients, the size of the seeds an individual produces depending on the environment. 7. And the alteration of leaf shape, size, and thickness. Leaves are particularly plastic, and their growth may be altered by light levels. Leaves in the light tend to be thicker, which maximizes photosynthesis in direct light, and have a smaller area, which cools the leaf more rapidly due to a thinner boundary layer. Conversely, leaves grown in the shade tend to be thinner, with a greater surface area to capture more of the limited light. 9. 10. Dandelion are well known for exhibiting considerable plasticity in form when growing in sunny versus shaded environments. The transport proteins present in roots also change depending on the concentration of the nutrient and the salinity of the soil. 11. Some plants, Mesembryanthemum crystallinum for example, are able to alter their photosynthetic pathways to use less water when they become water or salt stressed. 12. Phenotypic plasticity. It is hard to explain and predict the traits when plants are grown in natural conditions unless an explicit environment index can be obtained to quantify environments. Identification of such explicit environment indices from critical growth periods being highly correlated with sorghum and rice flowering time enables such predictions. 6. Additional work is being done to support the agricultural industry, which faces severe challenges in prediction of crop phenotypic expression in changing environments. Since exporting the global food supply are grown in a wide variety of environments, understanding and ability to predict crop genotype by environment interaction will be essential for future food stability. 14. Phytohormones and leaf plasticity. Leaves are very important to a plant in that they create an avenue where photosynthesis and thermoregulation can occur. Evolutionarily, the environmental contribution to leaf shape allowed for a myriad of different types of leaves to be created. 15. Leaf shape can be determined by both genetics and the environment. Environmental factors, such as light and humidity, have been shown to affect leaf morphology. 17. Giving rise to the question of how this shape change is controlled at the molecular level. This means that different leaves could have the same gene but present a different form based on environmental factors. Plants are sessile, so this phenotypic plasticity allows the plant to take in information from its environment and respond without changing its location. In order to understand how leaf morphology works, the anatomy of a leaf must be understood. The main part of the leaf, the blade or lamina, consists of the epidermis, mesophyll, and vascular tissue. The epidermis contains stomata which allows for gas exchange and controls perspiration of the plant. The mesophyll contains most of the chloroplast where photosynthesis can occur. Develop wide blade lamina can maximize the amount of light hitting the leaf, thereby increasing photosynthesis. However, too much sunlight can damage the plant. Wide lamina can also catch wind easily, which can cause stress to the plant, so finding a happy medium is imperative to the plant's fitness. Genetic regulatory network is responsible for creating this phenotypic plasticity and involves a variety of genes and proteins regulating leaf morphology. Pheromones have been shown to play a key role in signaling throughout the plant, and changes in concentration of the phytohormones can cause a change in development. 18. Studies plant species Ludwigia arcuata have been done to look at the role of abscisic acid ABA, as L. arcuata is known to exhibit phenotypic plasticity and has two different types of leaves, the aerial type leaves that touch the air and the submerged type leaves that are underwater. ABA to the underwater shoots of L. arcuata, the plant was able to produce aerial type leaves underwater, suggesting that increased concentrations of ABA in the shoots, likely caused by air contact or a lack of water, triggers the change from the submerged type of leaf to the aerial type. This suggests ABA's role in leaf phenotypic change and its importance in regulating stress through environmental change, such as adapting from being underwater to above water. In the same study, another phytohormone, ethylene, was shown to induce the submerged leaf phenotype unlike ABA, which induced aerial leaf phenotype. Because it tends to stay endogenously within the plant when underwater. This growth in concentration of ethylene induces a change from aerial to submerged leaves and has also been shown to inhibit ABA production, further increasing the growth of submerged type leaves. These factors, temperature, water availability, and phytohormones contribute to changes in leaf morphology throughout a plant's lifetime and are vital to maximize plant fitness. Animals. The developmental effects of nutrition and temperature have been demonstrated. 20. The gray wolf, Canis lupus, has wide phenotypic plasticity. 21-22. Additionally, male speckled wood butterflies have two morphs, one with 3 dots on its hindwing, and one with 4 dots on its hindwings. The development of the fourth dot is dependent on environmental conditions, more specifically, location and the time of year. 23. In amphibians, Pristomantis mutabilis has remarkable phenotypic plasticity. 24. As well as Agallichnus callidrias whose embryos exhibit phenotypic plasticity, hatching early in response to disturbance to protect themselves. Another example is the southern rockhopper penguin. Rockhopper penguins are present at a variety of climates and locations: Amsterdam Island's subtropical waters, Kerguelen Archipelago and Crozet Archipelago's subantarctic coastal waters. 25. Due to the species plasticity, they are able to express different strategies and foraging behaviors depending on the climate and environment. 25. A main factor that has influenced the species' behavior is where food is located. 25. Temperature. Plant responses to temperature are essential among ectothermic organisms, as all aspects of their physiology are directly dependent on their thermal environment. Thermal acclimation entails phenotypic adjustments that are found commonly across taxa, such as changes in the lipid composition of cell membranes. Temperature change influences the fluidity of cell membranes by affecting the motion of the fatty acyl chains of glycerophospholipids. Because membrane fluidity is critical for cell function, ectotherms adjust the phospholipid composition of their cell membranes such that the strength of van der Waals forces within the membrane is changed, thereby maintaining fluidity across temperatures. 26. Diet. Plasticity of the digestive system allows some animals to respond to changes in dietary nutrient composition. 27-28. Diet quality. 29-30. And energy requirements. 31-32, 33. Changes in nutrient composition of the diet. The proportion of lipids, proteins and carbohydrates may occur during development, e.g. weaning, or with seasonal changes in the abundance of different food types. These diet changes can elicit plasticity in the activity of particular digestive enzymes on the brush border of the small intestine. For example, in the first few days after hatching, nestling house sparrows Passer domesticus transition from an insect diet, high in protein and lipids, to a seed-based diet that contains mostly carbohydrates. This diet change is accompanied by twofold increase in the activity of the enzyme maltase, which digests carbohydrates. 27. Acclimatizing animals to high-protein diets can increase the activity of aminopeptidase N, which digests proteins. 28-34. Poor quality diets, those that contain a large amount of non-digestible material, have lower concentrations of nutrients, so animals must process a greater total volume of poor quality food to extract the same amount of energy as they would from a high-quality diet. Many species respond to poor quality diets by increasing their food intake, enlarging digestive organs, and increasing the capacity of the digestive tract, e.g. prairie voles. 33. Mongolian gerbils. 30. Japanese quail. 29. Wood ducks. 35. Mallards. 36. Poor diets also result in lower concentrations of nutrients in the lumen of the intestine, which can cause a decrease in the activity of several digestive enzymes. Animals consume more food during periods of high energy demand, e.g. lactation or cold exposure in endotherms. This is facilitated by an increase in digestive organ size and capacity, which is similar to the phenotype produced by poor quality diets. Common degus, Octodon degus, increase the mass of their liver, small intestine, large intestine and cecum by 15-35%. Increases in food intake do not cause changes in the activity of digestive enzymes because nutrient concentrations in the intestinal lumen are determined by food quality and remain unaffected. Intermittent feeding also represents a temporal increase in food intake and can induce dramatic changes in the size of the gut. The Burmese python, Python malurus bivitidis, can triple the size of its small intestine just a few days after feeding. 37. AMY2B, alpha amylase 2B, is a gene that codes a protein that assists with the first step in the digestion of dietary starch and glycogen. An expansion of this gene in dogs would enable early dogs to exploit a starch-rich diet as they fed on refuse from agriculture. Indicate the wolves and dingo had just 2 copies of the gene, and the Siberian husky that is associated with hunter-gatherers had just 3 or 4 copies, whereas the Saluki that is associated with the Fertile Crescent where agriculture originated had 29 copies. The results show that on average, modern dogs have a high copy number of the gene, whereas wolves and dingoes do not. The high copy AMY2B variants likely already existed as a standing variation in early domestic dogs, but expanded more recently with the development of large agriculturally based civilizations. 38. Parasitism. Infection with parasites can induce phenotypic plasticity as a means to compensate for the detrimental effects caused by parasitism. Commonly, invertebrates respond to parasitic castration or increased parasite virulence with fecundity compensation in order to increase their reproductive output, or fitness. For example, water fleas, Daphnia magna, exposed to microsporidian parasites produce more offspring in the early stages of exposure to compensate for future loss of reproductive success. 39. Reduction in fecundity may also occur as a means of redirecting nutrients to an immune response, 40, or to increase longevity of the host. 41. This form of plasticity has been shown in certain cases to be mediated by host-derived molecules, e.g. schistosomin in snails, Limnia stagnalis infected with trematodes, Trichobilharzia ocellata, that interfere with the action of reproductive hormones on their target organs. 40. In reproductive effort during infection is also thought to be a less costly alternative to mounting resistance or defense against invading parasites, although it can occur in concert with a defense response. 43. Hosts can also respond to parasitism through plasticity in physiology aside from reproduction. House mice infected with intestinal nematodes experience decreased rates of glucose transport in the intestine. To compensate for this, mice increase the total mass of mucosal cells, cells responsible for glucose transport, in the intestine. This allows infected mice to maintain the same capacity for glucose uptake and body size as uninfected mice. 44. Phenotypic plasticity can also be observed as changes in behavior. In response to infection, both vertebrates and invertebrates practice self-medication, which can be considered a form of adaptive plasticity. 45. Various species of non-human primates infected with intestinal worms engage in leaf swallowing, in which they ingest rough, whole leaves that physically dislodge parasites from the intestine. Additionally, leaves irritate the gastric mucosa, which promotes the secretion of gastric acid and increases gut motility, effectively flushing parasites from the system. 46. Self-induced adaptive plasticity has been used to describe situations in which a behavior under selection causes changes in subordinate traits that in turn enhance the ability of the organism to perform the behavior. For example, birds that engage in altitudinal migration might make trial runs, lasting a few hours that would induce physiological changes that would improve their ability to function at high altitude. 47. Air caterpillars, Grammia interrupta, infected with tachnid flies increase their survival by ingesting plants containing toxins known as pyrrolizidine alkaloids. The physiology for this change in behavior is unknown. However, it is possible that, when activated, the immune system sends signals to the taste system that trigger plasticity in feeding responses during infection. 45. Reproduction. Hatch rates for red-eyed tree frog tadpoles depends on predation. Red-eyed tree frog, Agalaichnus call-idrias, is an arboreal frog hylid that resides in the tropics of Central America. Unlike many frogs, the red-eyed tree frog has arboreal eggs which are laid on leaves hanging over ponds or large petals, and, upon hatching, the tadpoles fall into the water below. One of the most common predators encountered by these arboreal eggs is the cat-eyed snake, Leptodira septentrionalis. In order to attain, the red-eyed tree frogs have developed a form of adaptive plasticity, which can also be considered phenotypic plasticity. When it comes to hatching age, the clutch is able to hatch prematurely and survive outside of the egg 5 days after oviposition when faced with an immediate threat of predation. The egg clutches take in important information from the vibrations felt around them and use it to determine whether or not they are at risk of predation. In the event of a snake attack, the clutch identifies the threat by the vibrations given off which, in turn, stimulates hatching almost instantaneously. In a controlled experiment conducted by Karen Moore-Kenton, hatching rate and ages of red-eyed tree frogs were observed in clutches that were and were not attacked by the cat-eyed snake. When a clutch was attacked at 6 days of age, the entire clutch hatched at the same time, almost instantaneously. However, when a clutch is not presented with the threat of predation, the eggs hatch gradually over time, with the first few hatching around 7 days after oviposition, and the last of the clutch hatching around day 10. Karen Moore-Kenton's study further explores the benefits and trade-offs of hatching plasticity in the red-eyed tree frog. 48. Evolution. Plasticity is usually thought to be an evolutionary adaptation to environmental variations that is reasonably predictable and occurs within the lifespan of an individual organism, as it allows individuals to fit their phenotype to different environments. 49-50. If the optimal phenotype in a given environment changes with environmental conditions, then the ability of individuals to express different traits should be advantageous and thus selected for. Hence, phenotypic plasticity can evolve if Darwinian fitness is increased by changing phenotype. 51-52. A similar logic should apply in artificial evolution attempting to introduce phenotypic plasticity to artificial agents. 53. However, fitness benefits of plasticity can be limited by the energetic costs of plastic responses, e.g. synthesizing new proteins, adjusting expression ratio of isozyme variants, maintaining sensory machinery to